In this special tribute, Susan Galbraith, Executive Vice President of Oncology Research and Development at AstraZeneca, and David Reese, Executive Vice President of Research and Development at Amgen, reflect on José's legacy of bringing meaningful therapies to cancer patients and their views on the state of oncology R&D.
It's good to see you, Susan.
It's nice to see you as well, David. How are you?
I'm doing well, thanks. One of the things we were going to talk about today was José Baselga. I know you knew him, of course, extremely well. Just interested in your thoughts on what his legacy is, but the broader impact on the field.
Well, José is a giant in the area of cancer research. Over at the totality of his career, and I've known José for probably closer to 20 years than 15 now. So when I first met him, I was struck by the same things that many people are, which is about the level of energy, enthusiasm, and curiosity about everything to do with cancer and the passion for that. So I had lots of interactions over those time periods. Quite often, when I first met him, I was working at Bristol Myers Squibb, and he was helping to design a neoadjuvant study in breast cancer. And so just a lot of engagement on doing something that was innovative and different from what we'd had before. And I think, the impression that I got at that point, which again is still a lasting impression, was just of somebody who really wanted to push things to the limits and push people beyond what they thought that they could do, and into doing something that was different and that was going to actually make a difference. You see that in every period of time that he's spent in different pieces, but also during the time that he was with us at AstraZeneca, which was really only 2 years in the end, just over. But in those 2 years, he did have a huge influence. So why don't you tell me about your interactions with José?
It dates back quite a long time. He was, in John Mendelssohn's lab at Memorial Sloan Kettering, where he did his training. He was 2 or 3 years ahead of me in the sort of fellowship training cycle. I was in Dennis Slayman's laboratory at UCLA. So these were the earlier days of HER2. We got to know each other because of work that both laboratories were doing, and then intersected many times over the years after that. So, I knew José for probably 25 years, maybe just a bit more than that. And, as you said, everyone was struck by his energy. That's probably the single distinguishing quality, sometimes characterized as like the sun. It warms you for a while. If you're too close for too long, it might burn you a little bit. But that was, really driven by his passion for ultimately changing standards of patient care. That's the one thing to me that really came through and really, fit the definition of a physician scientist, someone who treated patients, someone who worked in the laboratory, and someone who really tried to bridge both of those worlds and bring the science of the laboratory to patients and ultimately across many classes of drugs in oncology. Many of the targeted therapies that we now rely on, he touched in one way or another.
I think he had a quite an unusual capability in both fields. There's quite a lot of physician scientists, but often there's a kind of pull in one direction versus the other. I think he was able to go between those worlds really quite fluidly, and see the relevance of scientific insight and how it then might make a difference for patients, and then pull it through in that way. I think that was really special. And one of the things that helped spur was the creation of a clinical fellowship program that we have, that we've actually now renamed in his honor as the Baselga Scholars. Because what he said he did it also at Memorial was get to know the people that were in the fellowships and be very stringent about the people that you're bringing in at that level, and then thinking about what you do to develop and stretch them I think this is really fascinating and actually very insightful way of thinking about the organization, the places where you can make the biggest difference to the quality of the talent and the development that you've got across the organization.
Yeah, that's such a critical piece because, some physicians and scientists, of course, their legacy is the work. Others, it is the cadre of people that they've trained and the generations of people that then carry the enterprise forward. And for some, it's both. And for me, Jose really fits into that latter category. And again, that's a small subset of the whole.
Yeah, it is. So I guess another question that we have is, what do you think the legacy is that Jose's left in oncology R&D?
I think it's much of what we've talked about, to me, but it's also that translational piece. Now we take, for granted that we want to translate ideas from the laboratory, bridge them into early clinical studies, and then really proof of concept and large scale testing as quickly as possible. But, we forget that paradigm is really, maybe a quarter century old. And so to me, providing models for that in some of the programs that he led, and then again, the institutional structures that supported it, to me, is the real legacy.
Yeah, absolutely. A champion of precision oncology, and also then understanding the very practical ways in which things need to work. I mean, again, go back to the HER2 example, an area that he's obviously worked on for many years. One of the key things that he's pushed us on right from the beginning was to make sure that we think about how we actually identify who is HER2 positive, what the lower bound of that could be, and how we do that. And that's another key piece about never accepting what's currently available and always pushing for something that is better. But I think as well, in the clinical trial space, in terms of clinical trial design, the speed at which you can design studies and the speed at which you can get them up and going. And then, always quoting, which is one clinical trial, one question. Just make sure that you understand what's the most important thing that you're actually hoping to learn from a particular study.
Absolutely. Simplifying clinical trials, understanding how to articulate a very crisp hypothesis so that the data you generate from that trial is actionable, usable, leads to decision making. He really, I think, pushed the entire field in that dimension.
Yeah, absolutely. I mean, just from a perspective of the other things that he's made a difference on here, obviously when he first came here, one of the things he said is wanted to help build a cell therapy group and increase the effort that we had in the epigenetic space. Is very influential in driving what I hope will be the next wave of programs. And actually I do think that even though a short period of time, it's made a huge difference to the advances that we can make in that space.
Yeah, that's great. Well, obviously, to me, Jose would be, thrilled about where we stand, in the midst of a technological revolution that I really think, in the coming decades will revolutionize cancer care, and well beyond that, actually many aspects of medicine. But let me ask you, I'm curious, what excites you? What technologies are you, really interested in at this point?
Well, there's as you say, I think you've already said that there's a potential for a revolution in how we treat cancer coming in the coming decade, and I think it's because some of the technological advances are enabling some scientific advances. Some of the technology that are enabling insights into potential new epigenetic targets are things like ATAC-seq, single cell sequencing, some CRISPR screening, etc., and the ability to then identify targets you can then do something about, because there's a range of those that come out of the CRISPR screens that would have been in the previously undruggable class, but with technologies like accessing protein degradation through things like PROTACs, etc., molecular glues. And I think it's incredibly exciting about what might be possible for us to do in the next 10 years.
Yeah, to me, we really are, at what you might characterize as a hinge moment driven by technology. part of the way I like to think about this is that we're entering the era of human data, from genomics, other omic technologies, protein structures. 6 months ago, we didn't know most protein structures. Then one day we woke up and 350,000 had been deposited into the public domain, 250,000 human protein structures, 100,000 from model organisms. We're now starting to tackle the protein-protein interaction problem. All of that on a molecular level, then clinical trials data, efficacy data, safety data, all of the associated biomarker and translational data. And now, of course, a tsunami of real-world evidence and real-world data coming at us. But all of that actually has in common that it is data derived from human tissues, human cells. So, the ideal model organism to study, the human, and to me, the sort of thrill and challenge of the next decade or two will be, how do we aggregate all of these data, put it together, and then crucially analyze it so that we get actionable, usable solutions. There's great promise. I really think we're on that cusp. It's going to require a gigantic ecosystem, of all of us to make a reality. But, we can now, we have line of sight into what a lot of the technical challenges are. We can see our way to solving them. There's a lot of hard work, and sweat that's going to be involved in the coming years. But to me, we're really on that threshold.
Yeah, no, I share your optimism and enthusiasm about the potential that we've got. I actually think we're already starting to see some of those insights come through that are making a difference. I think the other thing that we haven't talked about just yet, that I think that has the potential to increase the proportion of people that can be cured from cancer, is the technological advances that enable us to detect cancer at an earlier stage. And I think the technological challenge of identifying people earlier is something that I'm very optimistic about, using circulating tumor DNA as well as better imaging, but predominantly going beyond where you can detect cancers by imaging. I think that's a fundamentally important part of what we're doing. And it involves doing something that Jose would have encouraged us to do, which is going to the white spaces, also help to form what the new endpoints are going to need to be in those settings, deliver the data that will help convince investigators and regulatory authorities that it's possible to think about drug development in those spaces at an earlier point.
Yeah, exactly. So I think it opens up an entire spectrum, from the very technical in terms of the characteristics of the molecular assays, through how you conduct the trials, through how you actually understand what you're seeing and have something that is now met an approvable regulatory threshold without taking 20 years to do the entire experiment. Well, one thing the pandemic taught us is that, the three legs of the stool, this stool, when they're working together, industry, academia, and government can move very, very quickly. We should never forget the example of getting vaccines roughly a year after development started in the face of a global pandemic. That tells us what's attainable. That tells us what the art of the possible is. And to me, it's on us to say, how do we apply that to other areas that are huge public health burden? Oncology, cardiovascular disease.
Yeah, no, I mean, the speed of being able to get studies up and going, was incredible. I'm actually very proud of the efforts that we've had in terms of the vaccination. The ability to deliver vaccines to multiple different countries around the world has been built off that very spirit. And I do think I absolutely agree with you that shows what is possible. But that's just one disease. Actually, if we applied that same approach to everything that we could possibly do for cancer and how we could collaborate in terms of bringing that forward, I think we could make a huge difference in a much shorter period of time, particularly in the areas where we need the collaboration, which is in, as we say, the endpoint design, etc. There's an opportunity for us to do that.
Yeah, absolutely. Well, one of the technologies we're really interested in, to bring to bear comes back to a word that you used earlier, which is undruggable. And, we know that 80 or 85% of the targets of interest right now are not druggable or tractable, given current technologies, the therapeutic modalities that we've got. And so to me, the next few decades will be devoted in large part to making the undruggable druggable. Now, we have a recent example in drugging KRAS, specific mutation, G12C. We were the first across the finish line, but there are 8 other molecules in the clinic. That took 40 years as a target for us to understand how you actually developed an inhibitor. But as the technology evolved, you see then many parties starting to move quite quickly. So how do we make the rest of that genome that's currently undruggable druggable? One thing that really excites me are what we call multi-specific molecules. For really the century and a quarter that this industry has been in existence, we have focused on one drug, one target, either turning it off usually, or sometimes trying to turn it on or agonizing it. Well, I think we're now entering an era, and of course we've got examples of this where you're going to have molecules that do more than one thing.
Yeah, I think the advances in this space are happening at pace, and it's great to see that we've actually got drugs like KRAS G12C directed therapies progressing so quickly. But we're really only starting to scratch the surface of that particular important target. So there's no question, I think, that a lot of what we think are difficult to target, I would call them difficult to target rather than impossible to target,
right,
these days. But we'll continue to make progress in that. I completely agree. I think the other piece that we haven't talked as much about yet, but is really important, is how we bring some of these pieces of the puzzle together. And combinations are likely to be important to actually get a higher proportion of patients that can achieve cure. And the way we've progressed with combinations has been much slower to date than I would like to see. So I think there's an opportunity as well to think about some of the studies that we design to make sure that we can facilitate more rapid progress of those combinations. And these human-based model systems that we talked about, I think are a key part of triaging the key combinations that we need to do, because quite often in the preclinical models, we've looked at the combination for efficacy, but we've not spent as much time looking at the potential for combination for toxicity. And I think in the human-based models, we've got a better opportunity to make sure that we're actually prioritizing those combinations that are going to drive an improvement in the therapeutic index, which I think is a critical component of what we need to do.
I think the pandemic here also taught us certain things. The most informative trials were some of the platform trials that were run that incorporated multiple agents, dropped those that weren't working, took forward the winners. There's a big example there. Can we set up ongoing platform trials in certain diseases, for example, that are almost evergreen and perpetually investigating new regimens? I mean, if not now, when can we ever be this creative, given what the pandemic has taught us again about the art of the possible?
Yeah, it's a good point. So one of the questions I think that's important for us to think about is what you think the future of cancer treatment is going to be if you sort of look ahead 10 years. Do you have any thoughts for that?
I think you're going to see, an absolute proliferation of modalities from many new targeted therapies that'll be built on the back of, the structural biology revolution that's unfolding in front of us, and the corresponding revolutions in both small molecule and large molecule engineering. And when you can bring into the clinic many more molecules because you're able to generate them for a fraction of the cost of what you're able to do. It actually allows us to re-envision the sort of early development enterprise as well. Instead of one drug against a target moving through, it either succeeds or fails, and then we move on to backup molecules if it fails. What if you could put a dozen molecules in the clinic at the same time and very quickly start to advance the winners, for the same, using the same number of patients and at the same cost? That's a game changer. And it's no longer pie in the sky. It's not quite here yet, but in 10 or 15 years, can it be here? Absolutely.
So yeah, that's really interesting. So one of the things I think that we really need to sort of focus on is how we increase the long-term survival and cure rate. And I think there's some fundamental principles that we can use that are already tried and tested, frankly, but we can use now with the tools that we're getting now to help build that. And just the recent example of innovation in HER2-driven antibody-drug conjugates is just one example. So I think putting those key things together is a really important way in which we're going to improve outcome. And then I think actually identifying patients at the early point of the disease and putting them into trials early is a fundamental thing which will accelerate that long-term survival outcome. The other thing that I think has really been helpful and will continue to be critical is thinking about it from the individual tumour strategy perspective. So the way I think about it, you've got kind of mechanism of action approach to cancer treatment, and then you've got what's relevant within the specific tumour settings, and you've got to be able to marry those two views together to help drive the right questions being asked in the right clinical trials at the earliest point. So those are key elements that we have in terms of our strategy, and things like using ctDNA as to identify patient populations, ways to accelerate that. So for example, we're using ctDNA rises in patients that have been on previous treatment regimens to identify the patient population most likely to benefit, test it in that setting with the new drugs that are coming out. I think these kinds of things can be disruptive and accelerate our pace of change for cancer treatment.
I think we have to erratically expand the clinical trials enterprise. It remains the case that only 4 or 5% of patients with advanced cancer, for example, enter a clinical trial. So, to me, for sure, Jose would be saying, all right, how do we think holistically here? How do we think across this entire field about how we get more patients on clinical trials? Because that actually may be the rate limiting factor in terms of how quickly we can advance, and how do we reach patient populations that have historically been not well represented in oncology trials. To me, two huge big tasks that I think would have been top of mind for him.
Yeah, well, we were already pushing on the diversity aspect in clinical trials, and there's a couple of things that we need. One is the democratization of access to the information about what the best approaches are, and how that information gets basically cascaded much more effectively across the broader patient population and the physicians who are treating them. Because let's be clear, as the avalanche of data comes within oncology, it becomes challenging as an oncologist and a community oncologist to be on top of all of that information. So digesting the data and understanding what's needed there, I think, is really critical. There's a real emphasis on quality as well. Quality in every aspect, quality of the science, quality in the clinical trial, quality in the questions that we're driving. Jose was relentless in pursuit of doing things well, absolutely relentless, and very cognizant all the time of the legacy that Jose does represent, and making sure we live up to that, because he has high expectations of all of us, I know.
Yeah, no, I think we approach this with humility because we know that there are formidable challenges, but also the sort of relentless optimism that all of the breakthroughs we see around us, from the atomic level through clinical trials methodologies, are really bringing to bear on this field. So I look forward to working with you on all of this.
All right, thank you. It's good to talk. Take care.
Take care.